So I'm Célia, and I'm gonna talk about the high-throughput sex sorting of transgenic or non-transgenic male larvae for genetic control of Aedes mosquitoes. So this comes in the frame of the sterile insect technique and genetic control methods in general that rely on the releases of large amounts of sterile males. And currently, one of the main bottlenecks is the sex separation that is operated at the pupal stage using mechanical methods such as metal sieves or glass plates, as you can see here. These methods are very error prone and have a low recovery rate. So that's the main thing we wanted to improve in our project. We wanted to do something similar to what has been done in Anopheles mosquitoes, which is genetic sexing strains tagging the Y chromosome with a fluorescence protein. So only the males were fluorescent. And then you use a machine that is called the COPAS that sorts the neonate larvae according to their fluorescence. The COPAS looks like this when you open it. You put your sample in the sample tank. They go through the first pipe that is here in front of a laser where the fluorescence is excited. And depending on the selection that you've made on your software, they're either sorted or discarded. In Aedes mosquitoes, we cannot link a fluorescence marker to a sex chromosome because there are no sex chromosomes sensu stricto. There are sex loci named small m and big M that are located on the first pair of autosomes. Small m is the equivalent of an X chromosome, and big M is the equivalent of a Y chromosome. So females are small m, and males are small m, big M. In the big M locus, there is notably the Nix gene, that is the masculinization gene. In Aedes aegypti, we have an aligned genome, which is a good starting point. And what we know is that outside the sex loci that are quite small, under 2 million base pair, there is a 68 million base pair region that encompasses the sex loci that do not recombine. So if we link a fluorescence marker to this region, it should be inherited with the sex loci. So we targeted a gene that is located in the middle of the non-recombining region at 9 million base pair from Nix when you're on the big M bearing chromosome. So this is this gene, and that's quite funny because this is a mucin. And we edited the gene, but without knocking it out by CRISPR-Cas9, and we obtained several insertions, several that were big M linked and several that were small m linked. The one that you can see on the picture here is a big M linked insertion because all the males are green, the red fluorescence being the transgenesis marker. Both lines that we selected for going further are suitable for COPAS sex separation. You can see here that the big M linked line has a nice sex separation because the males are fluorescent and the females are not fluorescent. And with the small M linked line, it's a little bit harder because it either they have one copy or two copies, but still you can make a very nice sex separation using the COPAS. In Aedes albopictus, the story was much harder because we do not have an aligned genome, so we can't just do targeted editing because we do not know what to target. So we wanted to try our luck with piggyback random integration, and we followed more than 120 separate insertions. And the best sex linkage that we got is a small m linked insertion that recombines at 0.1%. This insertion is quite good for sex separation. We just had to modify it a bit, and now we get a nice sex separation with polyubiquitin and GFP marker. And for the big M locus, what we observed in another study is that piggyback has a tendency to home near a sequence that is within the plasmid. So what we did is that we built piggyback plasmids bearing a fluorescence marker with a sequence of the partial sequence of the NIX gene. And this way, the plasmid is preferentially integrated in the neighborhood of the endogenous NIX gene. We obtained several big M-linked insertions, and we went further with that YFP1 that allows a good sex separation at the neonate stage. So these lines allow the direct sorting of transgenic males, especially the big M-linked ones. But as all countries do not allow the release of transgenic individuals, we also propose a scheme that would allow the sex separation of non-transgenics. So from this line, you could sort the non-transgenic females, and then from the other one, you can sort hemizygous transgenic males. You get an intermediary colony in which only the male is transgenic, and as it bears the transgene on its small m locus, then only his daughters will inherit the transgene. His sons will be wild type. So if you sort them, you get non-transgenic males. We did a bilot experiment both in Aedes aegypti and in Aedes albopictus, and here are the results for Aedes aegypti. In the first colony, we sorted 1,000 non-transgenic females. In the second one, we sorted 300 hemizygous males. And in the progeny, we obtained 66,000 larvae, out of which we could sort 30,000 non-transgenic males. We tested the different lines and the intermediary colony for their fitness. And what we observed is that the hatching rate is similar or higher in the tested lines compared to the wild type, both in Aedes aegypti and in Aedes albopictus. We also tested the big M-linked lines for their competitiveness against wild type males. And in Aedes aegypti, there was no difference, while in Aedes albopictus, we observed some reduction in competitiveness. And we tested the flight ability of the big M-linked lines and the non-transgenic males from the scheme. And what we showed is that they have the same flight ability as non-transgenic males. So with all these results, we wanted to see whether the method was operational or not. So we carried out a speed test to determine whether we could sort by COPAS at a high enough speed with good accuracy. And what we picked as an optimal speed would be around 60 larvae per second. That would allow about 70% male recovery with 0 to 0.1% female contamination, and 100,000 neonate male larvae could be purified in under 1.5 hours. So in conclusion, we developed 2 Aedes aegypti and 2 Aedes albopictus genetic sexing strains. We proposed the use of the direct genetic sexing strains for sorting transgenic males or the combined use for sorting non-transgenic males. Their fitness and the sorting speed seem in line with an operational use. There are some things that still remain to be improved and understood. In the big M-linked line from the Aedes aegypti strain, we observed some recombination at the 15th generation, so quite late, which suggests that the non-recombining region in which we landed is actually a reduced recombining region, but recombination is still possible. So if we want a better genetic sexing strain, we might want to go closer to the actual sex loci. Mass rearing and semi-field tests remain to be performed for getting rid of this here. and seeing if our strains are operational or not. With this, I want to thank my collaborators, Myriam Burbuchler, René Provetti-Olmo, and my collaborators and supervisor, Thierry Baldé, Jérémy Bouillet, and Eric Marois, as well as our founders. Thank you for your attention. If you have any questions, I'd be very happy to take them.